Assalamu alaikum second year. In previous two lectures, we have so far covered the components and functions of external and middle ear, and also the basic features of sound. And I told you about the role of tympanic membrane and the auditory ossicles in the sound transmission. Also, I hope that two of your university questions they are prepared for the impedance matching and tympanic reflex. Right. So, what are the learning objectives for today's lecture? I will give you the introduction to the bony and the membranous labyrinth. Then we will look into the physiologic anatomy of the cochlea and its functional units. I will tell you about the compartments of cochlea and how these are formed by two membranes inside the cochlea. We will have a look at the structure and function of the basilar membrane. Then I will also tell you the importance of the other membrane, which is called the REISSNER membrane. And I hope that one of your university questions that will be prepared from this lecture about the structure and function of the organ of CORTI. Um, then also I would like you to enlist the differences between inner and outer hair cells and know that what are the stereocilia. And finally, we will super focus on the cochlear innervation and its fluids, including the endocochlear potential, along with the mechanism for the generation of this endocochlear potential. So let's start with today's lecture. You see, it is about the inner ear, which is also called labyrinth. And in this labyrinth, uh, you see there is cochlea, which is the sense organ for hearing. I told you that our ear it performs dual function. One is hearing and the other is equilibrium. So the sense organ for hearing is cochlea, while that of the equilibrium that is vestibular apparatus. Now the labyrinth means the inner ear that consists of two structures, or you can say layers, the bony labyrinth and the membranous labyrinth. Right. Bony labyrinth is the series of cavities or channels which are present in the petrous part of the temporal bone. And membranous labyrinth that is situated inside the bony labyrinth. The space between the bony and the membranous labyrinth that is filled with a fluid which is called perilymph, or the other name for this fluid that is periotic fluid. Then we move on to the membranous labyrinth. Uh, the membranous labyrinth that is formed by membranous tubules and sacs, and it contains endolymph, right? The bony labyrinth, this was containing the perilymph, right? The space between the bony and the membranous labyrinth that contained perilymph, while the membranous labyrinth, it contains endolymph. Now, this endolymph, this is also called otic fluid. And a special characteristic of this endolymph is the level of the potassium ions which is present in it. And that level of the potassium ions, that is very high. Okay. Now, this membranous labyrinth, it consists of two portions. One is the cochlea and the other is vestibular apparatus. And I told you that cochlea is responsible for hearing, while the vestibular apparatus, that is for the equilibrium. Here, as far as hearing is concerned, we will focus on the cochlea. So, first of all, uh, we look at the physiological anatomy of the cochlea. You see that cochlea is a coiled structure. It is pea-sized and snail-shaped coiled structure, and it forms the hearing portion of the inner ear. This cochlea, it lie in the temporal bone, and basically it is a tube which is around 35 millimeter long, but that tube is coiled and it makes 2 and 3/4 turns. Mhm. Okay. Um, right. The cochlea itself, it consists of two structures, modulus and a bony canal. This bony canal, it winds around the modulus. And what is modulus? Modulus basically, it is the central axis, and this axis is in the form of cone. Uh, a cone which is formed by the spongy bone, right? Uh, so modulus basically is forming the central axis around which the bony canal, it winds, right? Now there is another feature that a bony, or you can say the osseous lamina, a spiral lamina, it projects from the modulus into the canal, right? So this is the structure of the cochlea, right? So we move on to the functional units of the cochlea, which are organ of Corti, stria vascularis, and spiral ganglion. The details of which I will tell you one by one. Um, if we consider cochlea as an instrument which produces electrical signals um, on encountering the sound vibrations, then this organ of Corti, that will be considered as a sensor, while the stria vascularis, it will act as a battery which will generate the voltage which is necessary to create the electrical signals, right? And that voltage, that is called endocochlear potential. Okay, so stria vascularis, that basically is required for generating the endocochlear potential. And spiral ganglion, you all know that a ganglion, that basically is a collection of the cell bodies of the nerve fibers. So this ganglion, this give off many nerve fibers which act as the electrical wires which will connect our cochlea to our central nervous system. Uh, in this figure, you can clearly see the functional units of the cochlea. This is the organ of corti. See, this is the cochlea. If we take a cross section of any part of cochlea, so inside the cochlear tube, inside the canal, we can see that here lies the organ of corti, and this is the spiral ganglion, which gives off the nerve fibers, and these nerve fibers, they are going to connect our cochlea with the CNS, right? And this part, the organ of corti, now this acts as a sensor. So if there are any changes, or if there is any stimulus in the form of these sound vibrations, then these sound vibrations, they will be detected here in the organ of corti. And this organ of corti, with the help of the hair cells, it will generate the electrical signals. In generating these electrical signals, one thing comes in handy, and that is the endocochlear potential. And for that endocochlear potential, this stria vascularis, this helps the stria vascularis because it generates the endocochlear potential. Because of this voltage, the electrical signals, they are generated in response to these sound vibrations, and then these signals, they are carried by the nerve fibers which are coming out of this spiral ganglion. You can see here this organ of corti, that it is mainly it is depending on the hair cells. These are the hair cells, the blue colors, and these hair cells, they have stereocilia. Now the movement of these stereocilia, that causes the generation of the electrical signals, which are picked by the sensory fibers, and then they are taken to the central nervous system. Right. So you can see that this cochlear tube, this has been enhanced here, and you can see that there are different cavities or chambers which are formed inside the tube, even the cochlear tube. So let's focus on these channels or cavities, or you can say the compartments. Right. So the cochlea, it has been divided into three compartments by two membranes. These compartments are called scala vestibuli, scala media, and scala tympani. Uh, Now, cochlea, that is divided into these three compartments with the help of two membranes. And which are those two membranes? Those are the vestibular membrane and the basilar membrane. Okay. The vestibular membrane, it is also called the Reissner membrane, while basilar membrane, it also has got a different name. You can also call it the membranous spiral lamina. Okay, it is different from the spiral lamina, which was the osseous spiral lamina. You see, there was a bony lamina. Lamina mean anything which is layer like. So the osseous lamina that was projecting from the modulus, and where the bony lamina, the osseous lamina ends, this membranous lamina it starts. It is the continuation of that bony or osseous lamina. Well, we are talking about the compartments of cochlea, which are three. That is scala vestibuli, scala media, and scala tympani. All three compartments, they are filled with fluid, but the fluid is different in different compartments. Scala vestibuli and scala tympani, they contain the perilymph, while the scala media, it contains the endolymph, right? Uh, and The basilar membrane that lies between the scala tympani and scala media. Okay, so let's have a closer look at the basilar membrane. Right. So here lies your basilar membrane between the scala media and the scala tympani. The middle one, that is called the scala media. Uh, I already told you that the basilar membrane, that is also called membranous spiral lamina, and it projects from the bony center of the cochlea, which is called modulus. I already told you. Uh, and see, this precisely, this membrane, it arises from the osseous spiral lamina to the spiral ligament that I have already told you. So we will have a look at the structure of this basilar membrane, because it is important that you understand this structure, that how the fibers of the basilar membrane, they are excited, they are moved, and their movement that results in the generation of the electrical signals from the hair cells, which are carried to the CNS. Right. So it is important to understand the structure of the basilar membrane. See, this basilar membrane, it's a fibrous membrane. It means that it is made up of the fibers. It contains 20,000 to 30,000 fibers. Uh, these fibers, they are stiff and elastic reed-like structures. You know what is reed-like? These are the reeds. Uh, these fibers of the basilar membrane, they they vibrate like the reeds of the harmonica upon the arrival of the sound vibrations. So this is how these fibers, they show movement, and their movement that results into the movement of the stereocilia, which are present on the hair cells, and this is how the electrical signals, they are generated in the hair cells, and then they are transmitted to the CNS. So the mechanical sound vibrations, they result into the generation of electrical signals by the hair cells, and hair cells, they are lying on top of this basilar membrane. So in whatever area the fibers of the basilar membrane, they are moved, they will result in the generation of the electrical signals in that particular area. in the hair cells of that particular area. And this is how uh, the basilar membrane that is involved in hearing. Now, the arrangement of these fibers that is very particular on this basilar membrane. At the base of the cochlea, the basilar fibers they are short and they are thick and stiff. Okay, while at the apex of the cochlea, the fibers they become thin and long. And this particular arrangement of the fibers and the difference in the structure of the fibers that is also important because this leads to the differentiation between different frequencies, because the fibers which are present at the base they are moved with a different frequency, while which present on the apex they are moved with a different frequency. So this is how the CNS it comes to know that what kind of frequency it has been heard by the ear, only by knowing that which part of the basilar membrane it has been stimulated. So this is how the basilar membrane is important in hearing. Um, it performs these functions. First of all, it causes the separation of the perilymph and the endolymph, because I already told you that it is situated between the scala media and the scala tympani. Scala media there is endolymph, and in the scala tympani and vestibuli there is perilymph. So it causes the separation of these two type of fluids because their chemical composition that is also different. Then it provides a base for the sensory cells. And which are those sensory cells? These are the hair cells which are present in the organ of corti. And you can see in this figure that the organ of corti that is lying over the basilar membrane. So it means that this basilar membrane that is providing a base for the sensory cells for hearing. It is providing support to the hair cells. And then a very important function it performs is the frequency dispersion along it. As I told you, that the fibers of the basilar membrane they show a different organization and different structure, uh, which is different at the base from that at the apex, and different kind of frequencies they cause the movement of these fibers. Only a particular frequency it will stimulate, or it will cause the movement of the fibers at the base, which will not cause the movement of the fibers at the apex. So this is how the basilar membrane it serves for the frequency dispersion along its whole length. Right. Wait. So you must keep in mind, um, this is the important thing to note, that the fibers of the basilar membrane they vary in length and thickness. Those nearest the oval window, means at the base of the cochlea, Those fibers are short and stiff, while those near the cochlear apex they are long and floppy. And I already told you that how the structure of the basilar membrane that is related to the excitation of the cochlear hair cells. That you see these sound vibrations they will cause the movement of the basilar membrane, and the fibers in a particular region of the basilar membrane, those will show vibration, they will be stimulated, and ultimately the hair cells, the cochlear hair cell present in that part of the organ of the corti, they will be stimulated, and then they will cause the generation of the electrical impulses. So this is how the mechanical vibrations, the mechanical sound vibrations, they will be converted into electrical signals. uh, the basilar membrane, it is very important. The other membrane, there were two membranes which were making three compartments in the cochlea. The other membrane, that is the Reissner membrane. The Reissner membrane, it is placed obliquely between the upper surface of the osseous spiral lamina and the upper part of the spiral ligament, right? This one is your Reissner membrane. And Reissner membrane is very thin. It means that it is very easy to cause the movement of this membrane. It is easily moved in such a way that it doesn't obstruct the passage of the sound vibrations from the scala vestibuli to scala media, right? Because of the thinness of the Reissner membrane, there is no hindrance in the transmission of the vibrations from the this uh, scala vestibuli to scala media. So whenever the vibrations they are going to be produced in the scala vestibuli, they will be transmitted to the scala media, and ultimately that will result also in the movement of the basilar membrane. uh, and since it is the membrane is very thin, for this reason the scala vestibuli and the scala media they are considered to be a single chamber. All right. Now, what is the importance of the Reissner membrane? Basically, it maintains a special kind of fluid in the scala media, which is required for the normal functioning of the hair cells. I already told you that the uh, scala media it has endolymph, while the vestibuli and the tympani they have got the perilymph, and the chemical composition that is different from each other. So that Reissner membrane that is also causing the that is also responsible for the separation of these two fluids, and then the Reissner membrane it maintains this endolymph in the scala media, because if the fluid uh, is mixed, or if the fluid is not endolymph in the scala media, then the and then it will mess with the endocochlear potential, which is the voltage which is necessary for the generation of the electrical impulses. And now we will look at the compartments of the cochlea. First of all, the scala vestibuli. Now, this scala vestibuli that lies above scala media. Here is the scala vestibuli. You see, this is the whole cochlear tube, partially uncoiled, and these are three chambers: the scala vestibuli, scala tympani, and scala media. So the scala vestibuli it lies above the scala media. Okay, and it arises from the oval window. Here you can see the oval window, and then this chamber, it follows the osseous canal up to the apex, and at the apex of the cochlea, it communicates this scala vestibuli, it communicates with the scala tympani through the helicotrema. Helicotrema, basically it's an opening. uh, so this scala vestibuli, a compartment of the cochlear canal, it goes all the way from the base to the apex, and at the apex, it communicates with the scala tympani through the helicotrema. Scala tympani, uh, that lies below the scala media, and it goes parallel all the way up to the apex, and then it is communicated by the scala vestibuli through the helicotrema, and then it starts at the helicotrema and then it ends at the round window, right? And finally the scala media, which is the most important one, and it is also called the cochlear duct. You can see here that this scala media, which is also called the cochlear duct, you can see it here, and the organ of Corti, this structure, this is organ of Corti that is situated in the scala media, right? So this scala media, this basically is a triangular compartment. uh, and this triangular compartment that is enclosed by the basilar membrane and the vestibular membrane, right? And this compartment, it ends blindly at the apex and at the base of the cochlea. It does not communicate with the scala tympani or scala vestibuli physically, right? So I told you that the cochlea, it has three functional units. One is the organ of Corti, then there was spiral ganglion, and an important functional unit of the cochlea was stria vascularis. So what is stria vascularis? Stria vascularis, basically, this is the stria vascularis. It is a highly vascularized and multi-layered tissue, and it forms the part of the lateral wall of the scala media, and it plays an important role in the cochlear hemostasis. How does it do it? Basically, it maintains the homeostasis of the cochlea by generating the endocochlear potential, because it maintains a unique ion composition of the endolymph which is present in the scala media. And there are different types of cells which are present in this stria vascularis. There are marginal cells, intermediate cells, basal cells, and so on. So keep in mind that the function of the stria vascularis is to maintain the cochlear homeostasis by generating the endocochlear potential, because for the sake of the endocochlear potential, it maintains a unique ion composition of the endolymph, which is different from that of the perilymph present in vestibuli and tympani. So the functional units of cochlea, those were spiral ganglion, uh, this stria vascularis, and the organ of corti. Organ of corti, that could be considered as body's microphone. It's a neuroepithelial structure. It is the receptor organ for hearing, located on the basilar membrane in one of the three compartments of cochlea. Right. So let's have a look at the structure and the function of the organ of corti. The organ of corti, that consists of thousands of sensory hair cells, which are attached to the basilar membrane and also to various supporting cells. Now, as far as function is concerned, the hair cells which are located in the organ of corti, they transduce the mechanical sound vibrations into the nerve impulses, and this is how the neural signals for hearing they are generated. Right. The functional components of the organ of corti. The functional components are the hair cells, which are inner and outer hair cells. Then there are supporting cells also, and the basilar membrane, of course, and the tectorial membrane. That is very important. So, what is this tectorial membrane? This tectorial membrane, that that forms the you can say the roof of the organ of corti, and you see that it is in contact with the processes, the stereocilia of the hair cells. It is in direct contact with these processes. So, it is in direct contact in such a way that these processes they are stimulated by the movement of the tectorial membrane in relation to the vibrations which are produced in the endolymph. So when the vibrations they are produced in the endolymph, which is present in the scala media, this tectorial membrane that shows movement. So when this tectorial membrane it shows movement, it causes the stimulation, it causes the the movement of these processes sideways, and this is how the hair cells they are stimulated by the movement of these stereocilia or the processes of the hair cells. That we will discuss in detail later. Okay, I tell you more about these hair cells. All right. You see, there are hair cells which are present in the inner ear, in the scala media, in the cochlear duct of the cochlea, and there are two types of the hair cells. There are inner hair cells, and then there are outer hair cells. Sensory nerve fibers, they are distributed around the cell bodies of these hair cells, and both inner and outer hair cells, they have short afferent and long efferent nerve fibers. So they have got afferent and efferent nerve fibers. The afferent nerve fibers, they are shorter, while the efferent nerve fibers, they are long, which are carrying the signals all the way to the CNS. Now, looking at the inner hair cells, they are flask shaped in such a way that they are broader than the outer hair cells, and these inner hair cells, they are arranged in a single row, and each hair cell, it has about 100 stereocilia. uh, Among these stereocilia, one cilium that is larger, and that larger one, that is called the kinocilium. Right. The inner hair cells, they are responsible for the sound transduction. It means that they are responsible for transducing the mechanical energy into the electrical energy. So basically, these are the primary cells which cause the generation of the action potential in auditory nerve fibers. Outer hair cells. So, the structure of the outer hair cells, that is very much similar to that of the inner cells. Although the mechanism of action, that is a bit different. Now, these outer hair cells, they show electromotility. And what does that mean, that they show electromotility? It means that these outer hair cells, they shorten during depolarization, right? And they are elongated during hyperpolarization. So mm, when they are shortened during depolarization, what happens, that they further pull the basilar membrane upward, and this is how they amplify the motion of the basilar membrane. So what happens in the electromotility of the outer hair cells, that they shorten during depolarization while they are elongated during hyperpolarization, right? Now this electromotility, that is due to the presence of a contractile protein which is called prestin. Uh, and this action of electromotility, it facilitates the movement of the basilar membrane and increases the amplitude of the movement of the basilar membrane. And this is how they serve the function of the sharpness of sound. They are responsible for the sharpness of sound by showing this electromotility. And I told you that what is this electromotility? That they shorten during depolarization and they are elongated during hyperpolarization. Okay? So this is called the electromotility of the outer hair cells. And uh, by performing this electromotility function, these outer hair cells, they provide an extra gain of 40 to 50 decibels to the hearing system. And for the same reason, they also perform the function of the tuning of the auditory system. right? So, on the basis of the all the data given about the inner and the outer hair cells, now you have to uh, draw the uh, column of differences between the outer and the inner hair cells. You can give the difference in the shape, as the outer hair cells, they are flask shaped, but the inner hair cells, they are broader than the outer one. So you can say that the outer hair cells, they are flask shaped, and the inner hair cells, they are goblet shaped. um, but a more important difference is that only 5 to 10% of the outer hair cells, they have got the sensory afferent supply, while 90 to 95% of the inner hair cells, they are sensory afferent fibers. They have got sensory afferent fibers. um, then if you look at the applied physiology, then the outer hair cells, they are more vulnerable to trauma. And what kind of trauma? The damage or the trauma which is caused by the ototoxic drugs. Your inner hair cells, they are more resistant. It means that they are less vulnerable to the trauma caused by the ototoxic drugs, right? So you have to make a column of the differences between the outer and the inner hair cells. Keep in mind that the outer hair cells, they allow us to hear low intensities, while the inner hair cells, they respond to medium intensities and also to the high intensities. And inner hair cells, they are more passive, while the outer hair cells, they perform an active function, and they also help the inner hair cells. Then you can also give the difference in the number of the outer and inner hair cells. You see that outer hair cells, they are more in number. They are around 12,000, while the inner hair cells, they are only 3,500, around 3,500. And since the outer hair cells, they are more in number, so they lie in 3 to 4 rows, while the inner hair cells, they form only a single layer. So that was all about the hair cells. Now we move on to the stereocilia. What are these stereocilia? Both the inner and the outer hair cells, they contain apical stereocilia. And these stereocilia, they are fundamentally important for the hair cell transduction. Now these stereocilia, they increase in length from the cochlear base toward the apex. The inner hair cell stereocilia, they are approximately twice as thick, and they are more, you can say they are more club shaped than the outer hair cell stereocilia. The more important thing is that there are lateral crosslinks between the stereocilia, which these links they attach the stereocilia to one another, right? So this is how these stereocilia they are connected to each other because of the lateral crosslinks. The outer hair cell stereocilia bundle it forms a V-shaped or you can say a W-shaped pattern whose angles they become more acute toward the cochlear apex and they form the that's enough anyway. So while the inner hair cell stereocilia they form a slightly you can say curved bundle. So the shape of the arrangement of stereocilia in the outer and the inner hair cells that is also differentiating. I hope that you get a picture of the hair cells and the stereocilia in your mind. So these were the functional units of the cochlea. I told you about the organ of Corti, and very important in the organ of Corti are the hair cells. And we mentioned that there are two types of hair cells, inner hair cells and outer hair cells. And you know the differences between the inner and outer hair cells. And then there are stereocilia, which are the processes which are present on the apices of these hair cells. Then I told you about the stria vascularis and why the stria vascularis that is important because it maintains the homeostasis of the cochlea by generating the endocochlear potential. Now, what is spiral ganglion? The neuron cell bodies which innervate the inner and the outer hair cells, they are located in the spiral ganglion. Now, the bony channel that connects the ganglion, it travel in a spiral direction toward the apex of the cochlea, and that is called the Rosenthal canal. Now, you see that approximately 90% of the spiral ganglion neurons they are so important that they are ensheathed by around 50 lamellae of myelin. Well, your spiral ganglion it gives off the nerve fibers which will carry the signals from your ear all the way to your CNS. Right, and then there are mm, then there are also some sporting cells in the inner ear. You see these sporting cells, and they have got different names, such as the phalangeal sporting cells. There are some outer marginal cells which are called Hansen cells. Then there are Claudius cells. Then there are border cells of Held, and so on. So, what are the rods of Corti? The supporting cells. Um, Now, these supporting cells, they are the inner and outer pillar cells. The pillars of the cells, they form series of arches, right? They form the series of arches which enclose a triangular tunnel, which is called Corti's tunnel. The basilar fibers, then the rods of Corti and the reticular lamina, they form a rigid unit and then they move as a single unit. Okay, the whole structure, this whole structure, this moves as a single unit. So whenever the basilar membrane it shows movement, this whole structure it moves as a single unit. So when this thing moves, it causes the movement, the basilar membrane it moves up and down, it will cause the movement of the stereocilia sideways. And when the stereocilia they move, they that movement result in the generation of the electrical signals. Now, once the signals are generated, how the signals they are carried to the CNS? Cochlea has got innervation. You see the cochlea that has got its nerve supply. It means that it is innervated, and the cochlea that is innervated by three types of nerve fibers. There are afferent fibers, mean the sensory fibers. Then there are efferent fibers also, which are, the bringing the orders or the information from the CNS back to the ear. And then there are autonomic fibers also. The autonomic nerve fibers, they do not appear to be present within the organ of the corti, but they are associated with the blood vessels and the nerve fibers of the modulus spiralemina, right? And there are the important ones are the afferent nerve fibers and the efferent nerve fibers. And I already told you that which type of hair cells they have got more afferent nerve fibers and which one they have got more efferent nerve fibers. Right, now we will have a look at the cochlear fluids. These cochlear fluids, these are the extracellular fluids. And these fluids are endolymph and perilymph. And they have, as you can see in the figure, that they have different chemical compositions, right? The perilymph that is present in the scala vestibuli and the scala tympani, both these chambers they are filled with the perilymph. And perilymph it is similar to the extracellular fluid in composition. And how it is similar to the extracellular fluid? Because it has a high concentration of the sodium ions, while scala media that is filled with the endolymph, right? uh, Now endolymph it contains high concentration of the potassium and it has low concentration of sodium. Why it has high concentration of potassium? It is due to the continuous creation of potassium ions by the stria vascularis into the scala media. So, uh, again, you will have to draw columns of differences between the perilymph and the endolymph. Uh, you see that the perilymph it resembles the extracellular fluid, while the endolymph that resembles the intracellular fluid. Then the location that is different. The perilymph that is present in the scala tympani and scala vestibuli, while the endolymph that is present in the scala media. And in the perilymph, the major cation that is sodium, while in the endolymph, the major cation is potassium. Now, I give you a very important difference in perilymph and endolymph, and that is that the perilymph, it has a potential, or you can say that it has a voltage of 0 millivolt, while the voltage, or you can say the potential of the endolymph, that is around plus 80 millivolts. So there is a difference of the voltage between the endolymph and the perilymph, and the difference is around 80 millivolts. This is very important in the understanding of the endocochlear potential. Now, what is endocochlear potential? Uh, you see, this endocochlear potential, that is also called the endolymphatic potential. It is the electrical potential which is developed in the fluids outside the hair cells. Basically, what happens? That an electrical potential, a difference, an electrical potential difference, a voltage difference of plus 80 millivolts, that exists between the endolymph and the perilymph with the positivity in the scala media. Okay, I already told you that the perilymph, it has a voltage of 0 millivolt. Endolymph's voltage is around 80 millivolts. So the voltage difference, the electrical potential of 80 millivolts, that is there between the endolymph and the perilymph, right? And the difference, the positivity in this difference, that is present in the scala media. It means the voltage of the scala media, that is uh, outside the cells, that is around 80 millivolts. And why there is this difference? This difference is basically due to the continuous excretion of the potassium ions by the stria vascularis into the scala media, right? So you see that there is a difference in the inside and the outside of the cells. There is a difference of the electrical voltage between two fluids even, and that difference is around 80 millivolts. Now we will see that how the endocochlear potential is generated and what is the cause for the generation of the endocochlear potential. Now you focus on this figure. The cause for the endocochlear potential, it is the difference in the potassium concentration of perilymph and endolymph. Because your endolymph, it contains way more potassium ion concentration. The value of the endocochlear potential, that is around 140 millivolts to 150 millivolts. You see, what is the mechanism? Uh, that the lower portion of the hair cell, that is dipped, or you can say that is bathed by the perilymph. Okay, while the head portion, this head portion, that is dipped by the endolymph. And you already know that there is a difference of plus 80 millivolts between the endolymph and the perilymph. Endolymph, it has a positive potential of endolymph. This has a positive potential of plus 80 millivolts. But inside the cell, ठीक है, because of the dipping of the basis into the perilymph, now Inside the hair cell, the electrical potential, that is the electrical potential which is, this is always present on the inner surfaces of the membrane. Now, inside the hair cell of the organ of the corti, this electrical potential, you can say the RMP, that is around minus 60 millivolts in comparison to that of the perilymph. So, here the electrical potential is minus 60 millivolts. Outside here, the electrical potential is plus 80 millivolts. So, if you make it from the minus 60 to plus 80, that will give you an endocochlear potential, that is the difference in the inside and the outside of the cell, that will become around 140 millivolts, because you take this minus 60 millivolts to the zero level, that makes the 60 millivolts, and then add another plus 80, so that will become 60 plus 80, that is equal to 140 millivolts endocochlear potential. So this is how you get 140 millivolts of endocochlear potential, which is the difference in the inside and the outside of the hair cell. See what a great difference between the inside and the outside of the hair cell. Why this greater value of endocochlear potential is required? The high potential difference sensitizes the hair cells so that the excitability of these hair cells that increases. It means that the spectrum is broadened basically. The spectrum of the sensitivity of the hair cells, that is broadened. Even if there is a very minor magnitude of the stimulus, which causes a very small change in the electrical potential between the inside and the outside of the hair cell, it will be detected. So, this is how the endocochlear potential of large value, it sensitizes the hair cells. That is just to achieve a greater excitability of the hair cells. And then also it increases the response of the hair cells, even to the slightest movement of the stereocilia. Well, I hope you understand this concept of endocochlear potential and why this endocochlear potential is important. Next. Right. So, that was your today's lecture. And if you have any questions, you can ask me directly. Next time, we will look at the mechanism of hearing, and we will start from the events which are involved in the hearing, and then we will move on to the sound transmission. uh, Now, this is enough for now. So, okay, take care.